Hello everyone, you are welcome on my YouTube channel Capsule. In the continuation of my previous video uh, on mechanism of drug absorption part 1, I, Dr. Vineet Kumar Rai, will tell you about the mechanism of drug absorption part 2 in this video. This will include the active transport mechanism as well as vesicular transport mechanism. As you all know that passive diffusion is a downhill process and doesn't require any energy in the form of ATP. But in the case of active transport, it is considered the uphill process and it requires energy in the form of ATP. Active transport mechanism are further subdivided into primary active transport and secondary active transport. Primary active transport involves direct ATP requirement because of the nature of uphill process. It helps transfer one ion or molecule only, that too in one direction, and that is why it is called as uniporter. And the best example of this kind is absorption of glucose. The carrier proteins are involved in this process. The carrier proteins works with the utilization of energy in the form of ATP, and these are of two types: ion transporter or ATP-driven ion pumps and the ABC transporter. In the case of ion transporter, these helps in transportation of ions into or out of the cell. So a classical example of this kind is proton pumps, and these pumps ensure the acidification of intracellular compartments. And the ion transporter that play an important role in intestinal absorption are organic anion transporters, which are used to absorb the pravastatin or atorvastatin like drugs, and organic cation transporter help in absorption of diphenhydramine like drugs. Now ABC transporter, these are responsible for transporting small foreign molecules out of the cell, and these foreign molecules can be drugs or toxins, and that is why these are called as efflux pump. These are only used to get something out of the cell, and a classical example of this kind is P glycoprotein, and it is responsible for pumping hydrophobic drugs out of the cell. So anyhow, the presence of large number of these proteins make the cells resistant to the drugs that are used in chemotherapy. Because uh, because of that, these are called as multi-drug resistant protein also. And ABC transporter present in brain capillaries and pump a wide range of drug out of the brain. So this is how ABC transporters work. Now the secondary active transport, as the name indicate, it differs from the primary active transport that require the direct ATP that utilizes direct ATP, and in this case the secondary active transport, it doesn't involve the direct requirement of the ATP. It takes advantage of previously existing concentration gradient. So based on that particular concentration gradient and based on the potential energy it has, this transport mechanism comes into the picture. Now the energy required in transporting one ion aids transport of another ion or molecule. It is called co-transport or coupled transport mechanism. It is seen either in the same direction or in the opposite direction. This process further subdivided into symport, that is known as co-transport, and antiport, that is known as counter-transport. In the case of symport, the movement of both molecule in the same direction happen. One potential energy of the concentration gradient of one molecule works for the second molecules, and with the first molecule, the second molecule goes to the other side. And example of this kind is sodium ion glucose importer that uses the potential energy of sodium ion concentration gradient. It helps move glucose against its concentration gradient. Another example is hydrogen ion coupled peptide transporter, that is also known as PEPT1, and it is implicated in intestinal absorption of peptide-like drugs such as lactam antibiotics. Now, the antiport, which is known as counter transport, it involves the movement of molecules in opposite direction, and the best example is hydrogen ion using the sodium ion gradient in the kidney. So, in this picture, I will tell you that how these transport mechanism work and active transport mechanism work. As I have already told you that it requires energy for transporting drug molecule to the other side. Here you can see that drug and carrier complex that is being available to the other side. After making the complex, this carrier molecule carry the drug molecule to the other side, and the drug molecule get dissociated. After getting free from the complex, this carrier molecule goes to the first site to carry another molecule. And this is the illustration of an active transport of the drug. Now, representation of the types of active transport, different types of active transport will be shown here. As you all know that uh, it works against the concentration gradient, and if the concentration gradient from the lower side to the upper side, so the active transport will be from the upper side to the lower side. So we'll see the first picture that is primary active transport, and it doesn't require any additional molecule to carry uh, the molecule of interest to the other side. And you can see here that the primary active transport of this molecule, and utilization of ATP to the ADP, and that is why it is the uphill process, and it is working against the concentration gradient, and the drug is getting available to the other side. So in the second case, another molecule is also working with a molecule of interest, and here this particular molecule is utilizing the potential energy or concentration gradient of this particular molecule and getting available to the other side with the help of this molecule, and this molecule is utilizing the energy. Okay, now you are seeing here that the same case, but here this molecule is utilizing the potential energy of this particular molecule to getting available to the opposite side, and this is how the secondary active transport work, and this case is called as import and this is called antiport. Now, active transport is an important process for the absorption of nutrient and several drugs. It differs from facilitated diffusion in several respects. So here the drug is transported from a region of lower concentration to the region of higher concentration in the active transport, and it works against the concentration gradient as we have already discussed. Now, absorption of ions against the electrochemical gradient, and that is why it is called as an uphill transport process. It doesn't get affected by achieving the equilibrium. When equilibrium is attained, only passive diffusion gets inhibited, and the process is faster than the passive diffusion. Energy is required in the work done by the carrier, and it can be inhibited by the metabolic poisons that interfere with the energy production process. So here, if the energy production process gets inhibited by any chemical or any poison, then obviously this active transport mechanism will be stopped. Some fluorides, cyanides, and dinitrophenol and lack of oxygen may stop the process. because of the poisonous nature, and endogenous substances like sodium, potassium, calcium, iron, amino acids, vitamins, and various other molecules can be transported by this mechanism. Now, some other drugs which are having the structural similarity with this, the carrier molecules can be transported by this mechanism, and chemotherapy is of particular interest today. And most of the and various chemotherapeutic molecules are getting available or getting absorbed by this mechanism. Some of the examples are absorption of 5-fluorouracil and 5-bromouracil via the pyrimidine transport system. The next example of active transport process is absorption of methyl dopa and levodopa via L-amino acid transport system. Another example is absorption of ACE inhibitor like enalapril via a small peptide carrier system. And that is why this transport mechanism is important in renal and biliary excretion of many drugs. And this process is used for the secretion of certain acids out of the CNS also. So after this discussion, I'll let you know about the comparison of active and passive transport mechanism. Suppose this is the gastrointestinal barrier, and this one is the gastrointestinal lumen, and this one is the systemic circulation, that is blood side, and drug will absorb from the gastrointestinal lumen to the blood side. So here, if at the absorption side the drug concentration is higher, then by passive diffusion it will go to the other side or systemic circulation by downhill transport. And if the drug concentration is lower at the absorption side, then it will move to the other side or into the systemic circulation by active transport, and that is the uphill transport. So that is why it requires energy, because it has to be reached at the upper side or up to the higher concentration level. As we all know that the stepping up requires more energy than stepping down. So that is the main concept behind it. Now, endocytosis is the other mechanism which is of minor importance, but it is also important. It involves engulfing extracellular materials. Engulfing means something eating or drinking of the cell. So it forms a saccule or vesicle, and it is called as corpuscular or vesicular transport. It helps absorption of drugs in solid state. It helps in cellular uptake of macromolecule nutrients. It helps in the absorption of fat, starch, vitamin A, D, E, K, B, and insulin. And drugs get absorbed into the lymphatic circulation directly, and that is why it is the best mechanism and that can bypass the first pass metabolism of the drugs. Now, endocytosis includes two types of processes. First one is the phagocytosis and second one is the pinocytosis. Phagocytosis means cell eating and pinocytosis means cell drinking. The cell eating absorption of solid particulates takes place, and in the cell drinking the uptake of fluid solute can be observed. The example of pinocytosis is the oral administration of Sabin polio vaccine, and large protein molecules, and botulism toxin by the pinocytosis mechanism. Now, in this case, you can see the biological membrane, and upper side the gastrointestinal lumen, and the lower side is the blood side, and drug will absorb from the upper side to the lower side. And here you will see that the macromolecule is being engulfed by the vesicle, and after engulfing it is being available to the other side, or you can say into the systemic circulation by forming vesicles. and this process is called as the cell eating. Now this is how the cell drink something, and this is the oily molecule, and it is being uh, engulfed by the vesicles, and this is the process of cell drinking, and this is how it is getting available to the other side. You can see here. Now this slide will include the combined all the mechanisms which I have discussed in my previous video and in this video, that is the active transport, passive transport, vesicular transport, uh, transcellular transport, paracellular transport. All these mechanisms will be shown in this slide, and this is of particular importance. And you can see here that this is the absorption site and this is the systemic circulation. Now you can see the different compartments. In the first case, this is the passive diffusion of the drug. The drug is getting available into the systemic circulation uh, by passive diffusion, and that is the downhill process, and it doesn't require any energy, and it is based on the concentration gradient of the drug. The higher the concentration of the drug on the absorption side, the faster will be the absorption rate. Now the next one is the vesicular, and it is also called as endocytosis mechanism, and phagocytosis and pinocytosis are the main mechanism of this kind. So next one is the pore transport. Here the drug gets absorbed by the pores between the cells. Next one is the carrier-mediated transport. It is the uphill process, and it is the kind of the active transport mechanism, and the carrier molecule requires ATP to bind some drugs, and after making a complex, it gets available to the other side, and after releasing the drug molecule into the systemic circulation, it goes to the first site to carry another molecule. Now facilitated diffusion, it is a kind of passive diffusion, and it doesn't require any energy. Here the intrinsic factor work to carry the molecule. Intrinsic factor help to bind the drug molecule with a carrier, and after binding with a drug molecule, this complex moves to the other side, and after releasing the drug molecule to the into the systemic circulation, it goes to the first site to carry another drug molecule, and this is how it works. Now finally, another passive diffusion process is ion pair transport, and here you can see the cationic drug is being complexed with an endogenous anion, and after making a complex of positive drug with a negative endogenous anion, the complex will be neutral, and this complex will have the lipophilicity, and due to the high lipophilicity of this complex, it will get available to the systemic circulation easily, and after getting available into the systemic circulation, it will release the cationic drug into the blood, and finally it will move to the other side to carry another cationic molecule. So this is how all these mechanisms work. Now some facts related to these mechanisms are shown here. A drug might be absorbed by more than just one mechanism, and cardiac glycosides are the best example that are transported by passive and active transport both. Vitamin B12 is absorbed by passive diffusion, facilitated diffusion, as well as endocytosis. And the transport mechanism also depend on the site of drug administration. Finally, in conclusory remark, the drugs with a high lipophilicity and molecular weight less than 400 Dalton are transported by passive diffusion process. The pore transport is suitable for the water-soluble molecules with molecular weight less than 100 Dalton. So pore transport is quite easy for the water soluble molecule having molecular weight less than 100. Now ion pair transport is suitable for the ionic drugs, cationic or anionic, at all pH condition, and it is facilitated by oppositely charged ion that form complex with a drug. Now drugs with a structure similarity with a carrier are transported by carrier-mediated transport, and macromolecules trapped in solid state and oily droplets are transported by endocytosis mechanism. So this is all about this video and this topic, that is mechanism of drug absorption. Now finally, in my next video, I'll tell you about the biopharmaceutical consideration in dosage form designing. So keep watching us. Thanks for watching.